Okay, so yeah, thank you for the introduction. Thank you for having me here today. Um, yeah, so I started my lab in January of 2021. So it's been kind of interesting getting started here. And I just feel like it's really the last couple months that we've gotten sort of up and running as far as advancing our research. So the talk that I'm going to give today is really a lot more skewed towards what I did as a postdoc. But I'm hoping that by talking about the system that I developed as a postdoc, you can get a sense for what some of the projects are in the lab and what some of the big initiatives that we're working on are. So my lab is sort of founded on the principle that bacteria are really good at colonizing things. So if you think about bacteria in the wild, these bacteria spend a great deal of time growing on solid surfaces, right? And so as you can see, this has a pretty significant impact on our everyday lives, particularly our health. But what I actually think is interesting about this behavior is that these cells that are growing on surfaces exist in a physiological state that's really different from the one that I learned about, which is basically E. coli growing in OB. So when we think about this, these cells, they have different behavior. They're resistant to antibiotic and phages. If you even look at them under the microscope, they look different. They grow different than cells that are growing in liquid. And so the theme of what my lab works on is trying to understand how bacterial cells achieve this surface adapted state. So we use this organism called Collobacter crescentus to study surface colonization. This is an aquatic organism. It was isolated from a lake, and it's pretty unusual. It has what's called a dimorphic life cycle. So this means that there's two cell types. And so every time one of these stalk cells at the bottom here divides, it releases a second cell type called a swarmer. And swarmer cells have pili and flagella, so they're modal. They can move through the environment, but they actually cannot replicate. And so in order for these swarmer cells to proliferate, they have to undergo this developmental program where they shed their flagella and pili and elongate a stalk before they can re-enter the cell cycle and divide. So one of the major things that we work on in my lab is a very specific event that can happen during this developmental program, where if the environmental conditions are right, these transitioning swarmer cells can make something called a holdfast. And so the holdfast is an adhesion, and it's extremely sticky. And so it allows these Collobacter cells to stick tightly to exogenous surfaces. So in my lab, generally speaking, we use holdfast production as a way to study how bacteria attach to and grow on solid substrates. And so my talk is kind of organized in two sections here. And the first section is based on the initial question that got me interested in studying Collobacter, which is related to the idea of how the holdfast is actually assembled. Because we knew from a few genetic studies that the holdfast was a polysaccharide, and it's a specific type of polysaccharide called a cell surface polysaccharide. These are incredibly widely spread in bacteria. Basically, all bacteria decorate their cell envelopes with these specialized types of carbohydrates, and these actually makes up the sort of outermost layer of the cell. So when we think about surface contact, this is the region of the cell that comes into direct contact with other objects in the environment. And so if you recognize some of these compounds that I'm showing you here, it's because they're virulence factors. So the infectious bacteria use compounds like O antigens, capsules, exopolysaccharides to kind of mask the cell surface during infections. And this is how these bacteria evade immune recognition when they're colonizing tissue. And so this is sort of a fundamental principle of how bacteria colonize things, is they modify their cell surface with these types of complex polysaccharides. And so when I started becoming interested in Collobacter, the question that I was really interested in was how these types of complex polysaccharides are actually assembled, and wanting to understand the mechanisms of the enzymes that do this. And so the advantage that we have in Collobacter here is that the holdfast confers this really powerful phenotype on the cells. Do I need to toy with the lights a little bit? Do you see, can you see any red dots on these slides? I'm gonna just turn the lights on. Okay. This one?
Yeah. Oh, yeah. It darkened it a little in the front.
Well, I can just describe what we're seeing here while we try and figure this out. So here we have 3 Colobacter strains. And yeah, here we go. So can you see these red dots now? Yeah. So I'm showing you the adhesive phenotype that you get from the holdfast by focusing on these 3 Colobacter strains that I'm showing here. We have wild type Colobacter, and we have a hyperadhesive mutant. This is a deletion of the holdfast inhibitor A gene. And we have a non-adhesive mutant where we've deleted this holdfast synthesis J gene. And so what you can see here is we're using this assay at the bottom called a crystal violet staining assay. So we're just growing these cells in liquid culture in a microtiter plate. We dump the culture, we wash the wells, and we stain the cells that remain attached. And so what you can see is that because the holdfast is required for adhesion, we can quantify holdfast production in individual mutants really quickly using this CV staining assay, right? So this approach of kind of picking tons of colonies and screening for mutants that have reduced adhesion was really the main way that people studied the holdfast when I started my postdoc. And for a lot of reasons, we knew that there were factors that were involved in holdfast biosynthesis that people had not identified yet. And so what I wanted to do was develop a way to sort of complete our understanding of the genetic basis for adhesion in Colobacter. And so I introduced a couple of new concepts to identifying holdfast synthesis genes. The first one was to say that screening individual mutants by crystal violet staining is a little bit laborious. And so what if we could actually just select for holdfast synthesis mutants? So what I did is I started growing Colobacter with cheesecloth. So basically what you do is you have this little patch of cheesecloth and you put it in these cultures, and when you shake them, what happens is the holdfast producing cells actually attach to the surface of the cheesecloth. And so if you look at the turbidity of the surrounding medium here, what you can see is that we're basically depleting holdfast producing cells as they partition into the cheesecloth. And so this is a way that we can enrich for mutants that don't have a holdfast in the surrounding medium. It's a much more efficient way to isolate holdfast synthesis mutants. The other approach that I brought in here is an approach for making what are called barcoded transposon libraries. And so this is a twist on a technique that's gotten really, really popular in bacteria in the last 10 years called transposon sequencing, or TN-seq. So essentially what we're doing here is we're making really big transposon libraries, and we're pooling all the mutants together in a population. So we have this population of mutants where we have transposon insertions spread at high density throughout the genome, and we can just map the transposon insertion sites in this population in bulk using next generation sequencing. So because my transposon is barcoded, when we do one of these mapping experiments, we basically correlate every insertion site to a unique barcode sequence. And then we can use that to basically count the abundance of each mutant in the population, right? So what we're doing in a typical experiment is we're counting the abundance of each barcode after some sort of experimental treatment, and we're comparing it back to the abundance of that barcode in a reference condition to understand how these mutations are affecting fitness, right? And the key to understanding this technique is really that it's a genome-wide quantitative approach. We're able to measure a phenotype for every single gene in the genome simultaneously. So what I did is I combined these two ideas of a selection and TN-seq, and I started making barcoded transposon libraries and subjecting them to a sequential passaging experiment in cheesecloth. And what I did is I just sampled this mutant population over time with TN-seq, sort of monitor the progression of this enrichment over time. So this slide here is showing you the 250 strongest phenotypes we identified in this initial experiment. So each line in this plot is a gene. And so I'm plotting how each one of these 250 genes affects adhesion as a function of time in this passaging experiment. And so what I'm going to show you here is how this approach is really useful for assigning function to new genes, right? Because if we have some unknown gene that has an adhesion profile that's really similar to a gene that we know what it does, we can just infer that it has a similar function. So this is what that looks like for holdfast genes. So here are two genes, 2360 and 2722, that really look like other holdfast mutants, right? And so when we go in and delete these genes individually, you can see that these 2360 mutants are non-adhesive, and these 2722 mutant cells have a severe adhesion defect, okay? So we can go in and we can look a little bit more detail at the cell to try and figure out what the defect is. So what we're doing on the top of this slide here in a second is we're staining these cells with a protein called wheat germ agglutinin. And this is a lectin protein that binds to a very specific sugar called N-acetylglucosamine. So when we stain these cells for glucanac, you can see these wild type cells are able to make a holdfast, but that these 2360 cells cannot. And so 2360 is not able to is required for holdfast synthesis, and we call this gene HFSL. These 2722 mutant cells were a little bit interesting because they're still able to make a holdfast. And so it took us a little while to figure out what was going on with these cells. But what we eventually did is what's called a slide attachment assay. So we just took a glass cover slip and we put it into a Caulobacter culture. We let the Caulobacter cells attach to the slide, and then we wash away the unattached cells. So in the wild type, you can see you have all of these cells coating the slide, and they're attached by virtue of making a holdfast, right? Of course, these HFSL cells, they can't attach because they don't make a holdfast. When we look at these 2722 mutant cells, we see that we're washing away a lot of the cells, but they're leaving behind all this holdfast material on the slide. And so this is characteristic of what's called a holdfast anchoring defect. This mutant is having a problem attaching the polysaccharide to the surface of the cell. We name this gene HFAE. So combining the information that we got from these two genes and a handful of others that we found with this approach, with what people had found over a couple of decades, finding mutants sort of one at a time with this pathway, we have a much more complete view of what the enzymes are required to make the holdfast are. And so this is a really complicated slide showing a polysaccharide biosynthesis pathway. And I want to talk in a little bit more detail about how this type of polysaccharide biosynthesis works. So there are a few different strategies that bacteria use to make these secreted polysaccharides. The holdfast is produced by a route called the WZY dependent pathway. And this pathway is sort of the most widespread in bacteria, and it also produces carbohydrates that are the most chemically complex. It's also the one that we know the least about. So when you want to make one of these extracellular secreted polysaccharides, you have this big challenge because if you're familiar with the bacterial cell envelope, this is a really thick, rigid, robust structure. It actually has multiple hydrophobic layers here. And so the challenge that these cells face is they have to produce these big, long, water-soluble chains of polysaccharides, but they have to do it with these resources in the cytoplasm. They have to use these nucleotide-activated sugars. And so they have this big challenge of solubility. And in the WZY pathway, the cell solves this problem by using a lipid carrier. So this lipid is called undecaprenyl phosphate. It's a special carrier. And in the first phase of this pathway, we have enzymes called glycosyltransferases that take these nucleotide activated sugars and use them to attach monosaccharides onto this lipid, okay? This culminates in the production of this really important intermediate called the repeating unit. And so the repeating units in different bacteria are incredibly variable. And that's because there's dozens of different glycosyltransferase families that these bacteria can use. Even within an individual family, the substrate specificity in terms of which sugars it binds to and attaches, these can vary widely. And so basically what you have is you have a system where every single strain has its own specific set of glycosyltransferases, and it produces a chemically distinct repeating unit. So this early phase of the pathway is really variable, but when we look at this assembly process that happens in the second phase, this is incredibly conserved throughout the bacterial kingdom. So what we have is we have an enzyme that flips this glycolipid to the outer leaflet of the cytoplasmic membrane. We have a polymerase that then takes blocks of these oligosaccharides and polymerizes them into a chain, and we have a complex that seems to secrete this thing to the extracellular space. So I think this model is really informative for talking about how sort of metabolites flow through the system and how the cell is able to get these things shuttled across the envelope. But the reason that I became interested in the system is that we don't understand these proteins very well at all. So even at the early phases here, these glycosyltransferase enzymes, we don't really understand in any detail at all how they're actually binding to these types of glycolipid substrates. And the big one is we don't understand the molecular basis for substrate specificity. We don't know what allows one enzyme to attach a specific sugar and not another one. And then the further you go through the system, the less we know. So when we get to this polymerization step that's really broadly conserved, we really don't know much about the mechanism of this enzyme. And so what I've wanted to do for a few years now is to reconstitute holdfast biosynthesis in vitro so that we can start to study the mechanisms of these enzymes. So with that in mind, when I go back to this model of what this polysaccharide production pathway might look like, this, when I presented this in my first paper, this was a very big overinterpretation of what was going on, because the problem that we faced was that we didn't know much about the carbohydrate itself. The only thing that we knew about the holdfast was that it had N-acetylglucosamine because we could stain it with with WGA. So really what this system looks like is this. Yeah, we've identified all the proteins, but we don't know what their substrates are. So if we want to reconstitute their activities in vitro, we don't know what to start feeding these enzymes. And so what I needed to do was I needed to find a way to extract the holdfast and analyze its carbohydrate content. And so on this slide, I'm really not going to go into a ton of detail here. This is a biochemical enrichment that I developed for the holdfast. It's based on the fact that we can stain the material with WGA. And so I grew up a ton of Colobacter and was able to isolate enough material to do carbohydrate analysis. So I want to talk through a little bit of what that analysis looks like. So here I'm showing you two sets of data. The first here on the top is called the composition analysis. Basically took the material and boiled it in acid to break it up into monosaccharides. And then we analyzed those residues by GCMS. These are the neutral sugars. So there's xylose, glucose, mannose. There's a little bit of methyl glucose. We have to run a different analysis for amino sugars, but we can confirm that N-acetylglucosamine is also present like we expected. This at the bottom here is called linkage analysis. So what we're doing here is a chemical trick where we're basically methylating these polysaccharides before we hydrolyze them. This kind of labels the free exposed hydroxyls. And then when we hydrolyze them, we can tell which residues were in linkages. So these individual sugars tend to be 4-linked here. This suggests that this is a beta-1,4 linked chain. We see these 4,6 linked and terminal residues that suggest that there's probably some branching happening. So this chemical work with the holdfast is really ongoing. We're trying to scale up these preps so that we can do some NMR and figure out the specifics of how these sugars are linked to each other. But in the context of thinking about reconstituting the system in vitro, this is a huge advance for us because, yeah, we learned a little bit more about what this polysaccharide might look like, but now we know the substrates that we need to reconstitute this process in vitro, right? We know what sugars we can feed to these enzymes to start building holdfast intermediates. And so I decided to remove some of this stuff from my talk, but this is essentially what we're working on right now. So we've started by saying, can we start to characterize the first enzyme in this pathway? This is data that shows that this first enzyme here called HFSE seems to specifically attach glucose to this lipid. And we've purified a number of other proteins in the system. So these are the 3 other glycosyltransferases that we think are acting downstream. And so eventually what we want to do is sort of scale up this reaction, purify this product, feed it to the next enzyme, purify that product, feed it to the next enzyme, and so that eventually we can start reconstituting polymerization in vitro, and we can really start to understand the mechanism of how these polysaccharides are built. So what I want you to take away from the first part of this talk is that we're trying to understand how bacteria are modifying the cell surface with polysaccharides. I want you to recognize that the role that genetics played in this process. We started out by saying, in order to reconstitute this system in vitro, we really need to know all of the enzymes that are required for this, right? Because if we're missing one step in the pathway, we can't get downstream here. And so that's sort of the inspiration for designing this saturating approach for studying holdfast mutants. But what I want to point out is that, everything that I talked about from this first screen was what we expected to see, right? This is what we wanted to do. We wanted to find holdfast synthesis genes. But sort of by accident, I found out that this is an incredibly powerful approach to find new genes, right? And so there were lots and lots of surprises that we found when we did this first screen. And so what I want to point out to highlight that, so I want to point out and talk for the rest of the time about these genes that are highlighted in blue here. So this group of genes has really interesting phenotypes in this passaging experiment. So they're actually giving negative fitness scores. What that means is that these mutants are being depleted more rapidly than the wild type by the cheesecloth. So they're hyperadhesive. When we look at the functional annotations for all of these genes, most of them are involved in the assembly of this machine called the flagellum. So if you're not familiar with the flagellum, this is a machine that drives bacterial motility. So here on the left, you can see a transmission electron micrograph of a bacterial cell, and you'll see this long filament extending away from the cell. So this filament is anchored to the cell through a transenvelope complex called the flagellar motor. And the motor, what the motor does is it just rotates this filament. And so when you get this filament spinning, it sort of acts like a propeller and it forces the cell forward through liquid. This is how these bacteria swim. And so this is the primary function for the flagellum, is to promote swimming. But when we saw this hyperadhesive phenotype in these flagellar mutants, it immediately made us think of this sort of secondary function that people have been noticing for the flagellum that involves something called surface sensing. And so this is a pretty simple model here that explains why bacteria want to do surface sensing, right? Bacteria don't just want to be adhesive at all times. They want to know if they're actually touching some sort of solid substrate before they commit to colonizing some sort of surface, right? And so they need a way to have a sense of touch. They need to know when they've encountered some sort of solid substrate. So what we and others sort of think is happening here is that the flagellar motor gets disrupted when you encounter a surface. And so I'm going to talk in a little bit more detail about how the motor works to try and give you a sense of what I mean there. So to understand how the flagellar works as a machine, you really want to focus on these complexes that are drawn in purple here, called the stators. So these stators are ion channels, and they use the proton motive force to turn this central part of the motor. And that's what gets this filament spinning and propelling the cell through liquid. So what we think happens when a cell encounters a surface is that the rotation of the filament becomes obstructed and the motor gets distorted in a way that the cell can sense as a cue to activate adhesion, right? So when we thought about this and we looked back at these hyperadhesive phenotypes, we started to think about this in the context of stalled assembly intermediates, right? So what we think is happening when we mutate these flagellar genes is that instead of them cells not having a flagella, they have misassembled flagella. And these misassembled flagella seem like they're basically mimicking the distortions that you see on the motor when a cell's on its surface. So these things are basically tricking the cell into thinking that it's on a surface. And so thinking about genetics, this was a really powerful phenotype because it's a gain of function phenotype. And by what I mean by that is that when we make these mutations, like deleting FLGH, we have a cell that now seems to be turning on a signaling pathway that leads to an increase in holdfast production. And so if we want to understand how that pathway works in more detail, we want to identify the genes in this pathway. We just need to find second site mutations that restore holdfast production back down to the basal level, right? So we needed to do a suppressor screen. And so what I did is I just made another barcoded transposon library, this time in the delta FLGH background, and I passaged it in cheesecloth to enrich for non-adhesive mutants. And so when you do this experiment, as you would probably expect, the holdfast synthesis genes are still the strongest determinants of adhesion in this flagellar mutant background. But we also identified about 40 genes that we think are downstream of the flagellum in terms of activating holdfast production. So we call these genes flagellar signaling suppressor, or FSS, and there's 40 of them. So this is a big initiative going on in my lab right now, is to understand what these genes are doing to activate holdfast production. But I'm going to spend the rest of the time sort of talking you through what we've learned so far. And I'm going to start by talking about these two genes here. So PLED is a cell cycle regulator. It regulates this Caulobacter cell cycle program. It's involved in stalk cell morphogenesis. MOTB codes for one of the stator proteins. So this is part of this torque generating unit that actually turns the flagellum. So what I'm going to show you on the next slide is that these two genes actually regulate holdfast production very differently. So to understand how this works, you need to think back to this idea about this stalled flagellar assembly intermediates, because we know a lot about how the flagellum is assembled in bacteria. And so what that means is that we can use reverse genetics and go in and disrupt specific stages of this process. And we can know sort of or predict what types of changes we'll see in the structure of the flagellum, right? So here, what we're doing is we're going to measure the effect of deleting PLED and MOTB in different flagellar assembly stage backgrounds, right? So here in the wild type background, we have a normally assembled flagellum. You delete PLED or MOTB, you have a really minimal effect on holdfast production. So now we're going to disrupt genes that are involved in the early stages of flagellar assembly. So we're going to break the process early on. You can look at these gray bars here and see that these single mutants are hyperadhesive. When we delete MOTB in these early stage flagellar mutants, we have minimal effect on holdfast production. But when we delete PLED, the cells are non-adhesive. So PLED is required for holdfast production in an early stage flagellar mutant. But we see a very different pattern when we start disrupting the later stages of flagellar assembly. So this time, again, when we look at these single mutants, the cells are hyperadhesive. But now if we delete PLD or MOP-B, we get a suppression of this hyperholdfast phenotype, right? So the effect of these two genes depends on where you're disrupting flagellar assembly. And these two different suppression patterns are telling us that there's two pathways here. So when we disrupt the early stages of flagellar assembly, we activate holdfast production through a pathway that requires PLD. When we disrupt the later stages, we activate through PLD and a second pathway that requires MOP-B. We can confirm that these are genetically separate pathways by knocking these genes out simultaneously. And so when you get rid of PLD and MOP-B simultaneously, the cells are non-adhesive, right? So this idea that there's sort of two parallel sub-pathways operating downstream of the flagellum has been really helpful for us because we have 38 other genes that we hit in the screen, and a lot of these genes are uncharacterized, right? And so being able to say, well, we can start by saying these genes should either be in the MOP-B or the PLD dependent branch of this pathway has been really useful for giving us a way to start going in and characterizing new genes that we hit in the screen. So for the rest of the time, I'm going to be talking primarily about genes that are uncharacterized that we found in this flagellar signaling screen. And I'm going to start by talking about this arm of the pathway that requires MOP-B. And we call this the mechanical pathway because we think it has a lot to do with how the flagellum functions as a machine. And so just to remind you about how this system works, we have these stator complexes. And if you look at the top view, you can see that these stators are surrounding this central rotor portion. And so the way this works, again, is these stators, when they start allowing ion flux, they sort of rotate in unison, and that's what drives the rotation of the filament, right? So what we're seeing as we start characterizing some of the genes that we hit in the screen is that it really is these proteins that are required to turn the filament. These are the same proteins that are required to sense filament obstruction. Okay, so to illustrate that point, I'm going to talk about two of the uncharacterized genes that we hit. We name these genes FSSA and FSSB. And so I'm going to look at the adhesion phenotype on the right-hand side of the slide. What we're doing here is we're knocking these genes out in different genetic backgrounds. You can see by looking at these purple bars here, when you knock out A and B in an early stage flagellar mutant, you have a minimal effect on adhesion. When you knock them out in a late stage flagellar mutant, you get this specific suppression of the hyperholdfast response. This is the same pattern that we see for MOP-B, right? So we can say these genes are involved in the MOP-B branch of the system. So what do these genes code for? So both of these proteins are predicted to have signal peptides for export from the cytoplasm. B has a TPR domain that's apparently associated with protein-protein interactions. So the functional annotations really aren't super helpful for these genes. But when we go in and we look at the motility phenotypes that you get when you knock them out, that's where we start to figure out what they do. So this is an assay called a soft agar spreading assay. This is a nutrient plate that has a low concentration of agar, and so it's sort of semi-solid. So what happens when we spot these cells into this plate is that if the wild type cells are able to sort of swim through the pores of this matrix and they're able to spread efficiently through the plate, you can see this delta FLGH, this strain is not able to swim. So here we have a delta FSSA and FSSB, and you can see that they have pretty severe spreading defects in this assay. So there's actually a lot of reasons why cells can have difficulty spreading through soft agar. So we went in and we looked in more detail at these cells. The first thing that we did is we looked specifically at flagellar assembly. So this is a method that we have for staining flagellar filaments. If you look at the wild type here, you can see the dye sort of cross-reacts with the holdfast because the holdfast is sticky. But we can also see the cells have flagellar filaments. Of course, this non-modal FLGH strain does not, and then we see flagellar filaments in these FSSA and FSSB mutant cells. So this motility defect is not due to a flagellar assembly defect, right? These cells still make a flagellum. The next thing that we did is we looked specifically at swimming in liquid. So we just took these cells and put them under a microscope. And what I'm showing you are maximum projections. So what we're basically doing is we're taking a video of these cells swimming in liquid and we're just overlaying all of the frames of the video on top of each other. It's a really crude way to track the motion of cells. But what you can see here is that this FSSB mutant is non-modal. It's actually interesting, this FSSA mutant is still able to swim in liquid. So starting with B, this is a really straightforward phenotype. This is a paralyzed flagellum phenotype. This is the exact same phenotype that you get when you delete MOP-B. These cells make a flagellum, they just can't turn it, right? A is pretty interesting. So it seems like it swims okay in liquid, but then when you challenge it with this higher viscosity soft auger, it starts to have motility problems. We have other data with A that I'm not going to show, but we're pretty convinced that what this phenotype for A is a sort of more modest form of this B phenotype. We think that deleting FSSA causes the cells to have a problem rotating the flagellar filament. So what we think these genes are is we think that they code for new components of the flagellar motor. And it's actually like a pretty exciting time to be thinking about the structure of the flagellum because, you know, with these advances in electron microscopy, there's like a higher resolution structure of the flagellum comes out basically every day, right? And so there, in a lot of bacteria, including Colobacter, people are starting to see these structures called collars that are involved in sort of connecting the stators to the outermost portions of the motor. And so we think A and B are really good candidates for being these collar proteins in Colobacter. Sort of more broadly, I think this is a really exciting thing that's coming out of our screen, is that we're excited to think about these new motor proteins that we're identifying as a way to think more about how the flagellum works. And I'm not just talking about surface sensing. I think we're also going to learn more about how this thing works as a machine when it comes to filament rotation. But the problem with this is that we've also started to see that like just taking this strictly mechanical view of how the flagellum works to control surface sensing is really not sufficient to explain all of the mutants that we identified in the screen. And so to highlight that, I'm going to highlight another uncharacterized gene. We call this gene FSSF. And so looking at the adhesion phenotypes here, I just want to briefly point out that when we knock FSSF out, we get this hyperadhesive phenotype. This probably should have been a clue to what this gene does, but you know, we missed it. Any case, what I really want to highlight here is that these suppression patterns, when you delete FSSF in an early stage flagellar mutant, we have no effect on adhesion. We delete the gene in the late stage mutant, we get the suppression. So again, this is the MoB pattern, right? This is this mechanical pathway of the system. So in this case, with FSSF, the functional annotations were actually really useful. So thinking back to the structure of the flagellum, I'm going to talk about another complex here called the C ring. This is the cytoplasmic region of this machine. And in E. coli and like basically all bacteria people have looked at, this is made up of 3 proteins called FlyG, FlyM, and FlyN. What's interesting about FSSF is that it's homologous to FlyN. It's not FlyN. Colobacter has a FlyN. It was discovered in the 80s, but it's like a second copy of FlyN. So we think that we sort of discovered a new C-ring protein here. And so, of course, what we did is we went in and we started looking at the motility phenotypes here. So this is another soft auger plate, and you can see that deleting FSSF, the cells are not able to spread through soft auger. Fine. What's actually interesting about this is when we go in and we start looking at the swimming behavior, we can see some differences. So first off, you can see that these cells are motile, they can swim. But what I really want to point out here is that the wild type cells have this really distinct swimming pattern where they swim straight for a while, then they bounce around for a little bit, then they swim straight, then they bounce around. You can sort of see that these FSSF mutant cells kind of have these meandering tracks. And so when we saw this, we knew right away what this was. This is a chemotaxis mutant. So this is an image of bacterial chemotaxis. This was actually taken here at UW. So this is a capillary tube full of aspartate, okay? And it's been inserted into an E. coli culture. So what you can see is these E. coli cells are congregating around the opening to this capillary. And so the reason for that is because they're attracted to the aspartate and they're swimming towards it. So this behavior where these bacteria are able to swim towards things that they like is called chemotaxis. And I want to tell you a little bit about how this works. So this is primarily based on studies in E. coli. E. coli is a little bit different than Colobacter, but the principles seem like they're the same. So E. coli actually has multiple flagella on every cell. And the default mode for these flagella is to spin counterclockwise, right? And so when all of these flagellar motors are spinning counterclockwise, the filaments bundle up into the superfilament and the cell goes forward, right? This is called running. But periodically, the flagellar motor can actually switch directions and switch from counterclockwise to clockwise. What that does is it breaks this filament up and causes the cell to stop swimming and do what's called tumbling, okay? So when you look at E. coli swimming in like a homogeneous solution, it has this sort of running and tumbling behavior. Where it swims straight, it tumbles, and then it swims off in a new direction. And the point about this behavior is this is called a random walk, because every time the cell tumbles, it swims off in a new direction at random, right? So this becomes interesting when you think about the cell in the context of a food source. So if you have some sort of attractant that's based over here, what you can notice about the swimming behavior here is that it's biased. When the cells are swimming towards the attractant, they tumble less. When the cells are swimming away from the attractant, they tumble more. And so this is called the biased random walk. And this is how bacteria actually move towards attractants. When we talk about the molecular biology here, this is driven by a kinase called key A and a receiver domain protein called key Y. When A phosphorylates Y phosphate goes and it binds to the C ring of the flagellum. So when phosphorylated key Y binds to the flagellum, this is what causes the switch from counterclockwise to clockwise, right? So when key Y is bound, this thing is spinning clockwise. Key A is associated with these big arrays of proteins called methyl-accepting chemotaxis proteins. And the only reason I'm talking about these is because these are the receptors for attractants, right? So when you have attractants present in the environment, they bind to these MCP proteins, they inhibit key A, and this lowers the concentration of phosphorylated key Y and inhibits tumbling. So this is the molecular mechanism for how attractants inhibit tumbling. So we became really interested in seeing these chemotaxis phenotypes in this delta FSSF cells. And so we wanted to go in and we wanted to look in more detail about how the cells are swimming. I told you that Colobacter is a little bit more complicated than E. coli. E. coli only has one key Y. Colobacter has 12 key Ys. But there does seem to be a key Y that behaves pretty similar to E. coli key Y. And so here I'm showing you a more detailed tracking analysis that we did with Jan DeFore at Michigan State. So here you can see that the wild type cells have this characteristic sort of run tumbling behavior where they're undergoing these sharp directional switches. And you can see that this key Y mutant has this sort of meandering running phenotype that we expect for a mutant that can't switch directions, right? So when we look at the FSSF mutant cells, again, we see that they're basically indistinguishable from these key Y mutant cells. And so this confirms that this is sort of a new chemotaxis mutant. It's pretty interesting because people haven't really been finding flagellar motor proteins that are required for chemotaxis. Usually the cells are just non-modal. And so that's sort of interesting in and of itself. But what happened is we started to go back and look in more detail about the data from the screen. And we saw that we've actually hit a handful of chemotaxis factors as flagellar signaling suppressors. This is the major key Y in Colobacter. And you can see it has this weak FSS phenotype. And then we can use this same sort of suppression approach to show that you delete this gene in an early stage mutant, you get no effect. You delete this gene in a late mutant, you suppress. And so again, this is a gene that's in the mechanical section of this pathway. So taking this back to this model of how this surface contact sensitive system works, we think that the chemotaxis system is integrated into this response. And so on the surface, that seems sort of not surprising because both of these systems, flagellar rotation and chemotaxis, target the flagellar motor. But the reason that I'm drawing it in this way is I want to point out the fact that we now have an entirely different sensory perception system here, right? We have this system that responds to surface, and we have this system that responds to nutrients, and they seem to intersect to control holdfast production. So that theme of sort of incorporating other sensory perception systems, we see that when we start looking at these genes on the PLD side of things. So before I show you some new genes, I want to talk in just a tiny bit of detail about what PLD does. So I told you this is a cell cycle gene in Colobacter, but it specifically actually controls the morphogenesis of stalk cells. So to understand what I mean, I'm going to show you these sort of classic electron micrographs of Colobacter. I want you to focus on these pre-divisional cells here. So in the wild type, this is what these cells should look like. We have a stalk at one pole, we have a constriction where the cell is dividing, and we have a flagellum at the other pole. This is what we should see. When we delete PLD, we get these weird cells where they have two flagella. They have a flagella at each pole. Sometimes you get these weird things where you have these stubby stalks with the flagellum sticking off of them. And this is because PLD is required for the morphogenic events that happen during stalk cell development, right? So we've found a handful of genes that seem to behave like PLD in the screen. And so I'm going to just highlight an example of two of those here. We call these genes FSSD and FSSE. So this time, again, we're looking at the adhesion phenotypes here. We delete these genes in an early stage mutant. This time we get suppression of the hyper-hold-fast response. We delete them in a late stage mutant, we get suppression. And so this is not the MOT-B pattern. This is the PLE-D pattern, right? These mutations suppress hyper-hold-fast response in any flagellar mutant. So we sort of wanted to see, do these mutants cause the same type of morphological defects that we see in PLE-D mutants? And we just didn't want to do it. So what we did is we developed a marker, fluorescently labeled marker for this protein of the flagellar motor called FlyM. We just tag it with Venus. And what you can see is that this localizes to the flagellar pole in Colobacter. You see these really beautiful sort of pre-divisional cells. You have a stalk here, you have a flagellar motor here, right? So what we've done is we've gone and we've deleted FSSD and FSSE in this genetic background. And what you can see is, I'm just throwing D here, but EE has the same phenotype. What you can see is that these cells are essentially indistinguishable from PLE-D cells. We have lots of pre-divisional cells that seem like they have two flagellar motors. We don't have very efficient stalk development happening here. And so we think that FSSD and FSSE are actually new cell cycle genes that seem to control the Colobacter developmental program. And I think that that's definitely interesting. Finding new cell cycle genes in Colobacter is always exciting, but I think it's much more interesting to continue building into this model because now we have another system that intersects with this surface sensing system, right? This system's a little different. It responds to internal cues instead of external cues, but nonetheless, this cell cycle signaling pathway seems to be intersecting with the surface sensing system through PLE-D, right? And so when we looked at this, we started to realize like, yeah, this is kind of how this works, right? We know about lots of different things that control hold-fast production. There are lots of different parameters in the environment that can influence whether these cells make a hold-fast. And then this is kind of how sensory perception works, right? You're kind of bombarded with all these signals and you need a system that's able to process that information and make a decision. And I think what's really cool about this system in Colobacter is that ultimately all of this information funnels and it converges on this transitioning swarmer cell to influence this sort of binary outcome. Does the cell make a holdfast or does it not? And so it's cool to think about this in the context of the fact that we've been doing genetics in the system for a number of years, and we found lots and lots of different hyperadhesive mutants. And so when you think about the way that delta FLGH works, you start to look at these and say, okay, each one of these mutants, they're activating some sort of signaling pathway that turns on holdfast production, right? And so we can use these as tools with our genome-wide screening approaches to understand the topology of this signaling network that I'm talking about, right? Because we can use some of these mutants to perturb specific parts of the system. And then we can go in and we can measure the adhesion phenotypes in the genome across this system during cheese-cloth passaging and basically just measure how each gene in the network is affected. And so one of the things that we're trying to get up and running in the lab is we're trying to develop computational approaches for comparing these fitness scores across different genetic backgrounds to sort of build unbiased maps for how this signaling network might work. And I think that this really touches on the point of what is so good about Colobacter, right? Because people have been studying surface colonization in bacteria for a really long time, right? But the thing about the traditional models for studying surface colonization is they're based on these things called biofilms, right? And so a biofilm is a mass of cells. And so forming a biofilm is a community behavior. It's many cells forming an aggregate over time, many generations, the cells divide. Colobacter does not form biofilms. Adhesion is entirely based on this sort of single cell phenomenon. And so we have this system where we have one single cell where it either makes a holdfast or it doesn't. And so we can measure this with our genetic system over and over and over again, trillions and trillions of times. And this gives us a really, really sensitive way to link the inputs and the outputs, right? We can discover really interesting things about how this system is regulated. And I think that that's what we're starting to see with this flagellar signaling screen is we're starting to see that we're able to pick up new mechanosensing genes that people haven't seen in other systems. And so I'm really excited to continue to apply these types of high throughput genetic approaches to understand something sort of fundamental about how signaling pathways integrate. So with that, I just want to go ahead and thank basically the members of my new lab here, a couple of my collaborators. Jan DeFore has been helping us with tracking cells in 2 dimensions. And then I've been collaborating with Parastu for years now to do carbohydrate analysis. I didn't really get a chance to talk about the in vitro work with the polysaccharides that we're doing with Jay, but yeah, just thanks again for having me, and I'm happy to take any questions that you have.
This colorvector doing with the rest of those key Y proteins.
Yeah, well, we hit 4 of them in our screen, and one of them is FSSE. So FSSE controls the cell cycle. So there's a key Y that controls the cell cycle, it seems like. Yeah. This is like a really open question in like bacterial signal transduction more broadly, is because like people have generally assumed that all of these key Ys bind to the flagellar motor, but like my model is that they don't. They go and they bind to other things, because I didn't go into details, but chemotaxis is basically 2-component signaling with like a ton of twists, right? And so I think that probably like these key Y-like proteins are either binding to other macromolecular complexes in the cell, or they're just influencing other kinase signaling systems. And I'm really excited about, because E. coli is really actually unusual when you think about bacteria. Like I think Pseudomonas doesn't have 12, but it has like 7 or 8 key Ys. Like most bacteria have way more than one. And I think it's an open question about sort of what these are doing. And that is something that, we're trying to think about what the other 3 that we hit are doing. Yeah, we haven't quite gotten there yet, but yeah, it's like definitely kind of one of the questions. Yeah. Yeah.
Yeah, this might be me not knowing enough about bacteria in the first place, but so it seems like chemotaxis is like part of the, like, could it contribute to holdfast behavior directly? It seems like we add certain things that like fish love, or there need to be that sort of stimulus.
Yeah, that's a really good question. So I'm going to actually try to remember to repeat the questions. The question is like, do we think that holdfast, right, or holdfast production is regulated directly by chemotaxis? Yeah, I mean, like it's a possibility. We know that like under certain medium conditions, like rich medium, the cells make lots of holdfasts, and then like in minimal medium, they make less. And this is a screen that we've thought about doing for a long time and just haven't. Is like, can we just grow these cells in different medium conditions and see which factors have become important in one and not the other? Yeah, I mean, it's a really interesting question, and I think the answer is like there's a lot of ways for nutrients to influence bacterial behavior. Yeah, I like ignored the really critical component of this system, and it's a second messenger called cyclic diGMP, and we sort of think of this as like the main positive regulator of holdfast production. But yeah, I mean, I think it's a possibility that we would be open to that, like sort of, I mean, would you envision it being like happening through a key Y-like protein, or just like a direct?
Yeah, that's what I was thinking. Would be something like that, where I could like do this directly.
Yeah. Yeah, I mean, it could be. That would be really cool. I think, yeah, given like the complexity that we're seeing here, like that could totally, yeah, that's an interesting idea. Yeah, thank you. Did you have a question about?
Well, sort of along the same line. So I guess the idea is that nutrient conditions in the calorie, everything's good, go fast with the day. Bacteria wants to stick to things and stay there.
Yeah, I think that is a concept that I agree with. It's just like, I mean, it's not unfortunate, it's just the way it is. Our data is sort of the opposite, right? Like you knock out key Y and you make less holdfast. So that really doesn't necessarily fit with how I would think about it, which is the way that you describe it. And actually what we see when we like flood these cells with nutrients is they tend to make more holdfast. But exactly sort of what that means in terms of chemotaxis, I don't quite know. And so I think the way that I'm gonna sort of try to address this is by saying that like genetics has been extremely good to us. Like we've been finding lots and lots of new genes, and the way that we do that though is like in bulk culture. And so everything is averaged out, and there's like a time element to this that we don't pick up, right? Like the cell doesn't like hit a surface and instantaneously just make a holdfast, right? And actually, like there's a lot of evidence for the fact that when the cells encounter a surface, they explore it for a little while. They see if they like it. And so I think that, you know, that's something that we're trying to get set up is like more sophisticated cell tracking systems to be able to look at how these cells are actually behaving when they're on a surface, when we knock out key Y or whatever. And I think that like that's probably going to wind up being the answer to like, what is it about the chemotaxis system that really like controls holdfast production is probably like, in my eyes, it requires both, like, right? It requires the cell to be on a surface and to like the surface. But this again requires sort of a different view of this process that's not based on bulk cultures and is based on like tracking individual cell behavior. Does that help?
So I have a second question related. Are the genes in operons?
For chem
Synthetic, you know, the holdfast synthesis or the.
Yeah, the holdfast genes are mostly in operons, but like, you know, and that's, you know, but most of the new genes that I found are not in the operon, and that's why people hadn't found them. Chemotaxis has a major operon, which like the key Y2, this major chemotaxis key Y is in the operon with like MCP and key A and these other things. But then like these other key Ys are kind of scattered around the genome. Some of them are just sort of orphans sitting there. D and E are like adjacent to each other. D is a protein. It's like a protein called KR. Chemotaxis is more complicated than I let on. And so some of these are like little chemotaxis modules that are missing parts, or like they're just scattered around the genome. But yeah, and then some of the others, like the flagellar genes are in operons, but then FSSF is not in the flagellar operon. It's somewhere else in the genome, right? Like, yeah. So I think that's like maybe a theme to a lot of the new genes that we found is like they just weren't sitting at the sites where people expected them to be.
So you might have a question in the chat.
Okay, while I get that up, Jake, do you want to?
Yeah, so like I was going back about glycosyl transferases. But you know, plant genomes have slew of glycosyl transferases, and it's really hard to predict what they do, and we carefully figure out one at a time, and that doesn't help us with the next genome that we're working on. So, you know, are there phylogenetic clues there about related to other bacterial glycosyl transferases? Can you make predictions about structure? That would be
Yeah, glycosyl transferases. I think that like this idea of actually predicting substrate specificity is like, I don't see that. Yeah, I mean, like, well, I'm not probably going to end up solving that problem, but like, I mean, right now we're thinking about it. Like, there are big bacterial families. This family is called GT2. I think plants have GT2 as well. It's pretty common. So like 2 of the holdfast ones are GT2. And like right now, I guess like my approach is really, you know, you have to go in and you have to do the in vitro experiments. I have not, I didn't talk about some of the in vitro work that we're doing, but like one of the cool things that we found is that the book on these enzymes is like most enzymes, like, oh, they have one substrate and like they make one product. But then when we go in vitro and we actually test like a panel of substrates, it's like now they're turning over 5 different substrates, right? And so I think in terms of specificity, I don't know that we'll ever get to a point where we can predict function as far as like that level of detail. But I think, yeah, I mean, I have a really biochemical approach, which is like, I just want to go in and see what they can actually do. And then, you know, maybe if we can find some rules later on.
So then do you think that you're sort of 4 or 5 steps or whatever it is, do you think that those are necessarily each doing a different committed step of that, or are they each contributing to maybe a mixture of?
We don't know, and that is another thing that we've seen sort of hints of in vitro. Like the system that we have is like really been slow to set up, but like, yeah, like we've seen things like we predict that this enzyme is downstream, but it reacts with, you know, the enzymes will react with the same. The other thing that we've seen that's kind of surprising is like we've seen multiple turnover. So like we think like, oh, these early glycosyltransferases, they add one sugar, but like we have pretty good evidence that one of these can actually attach two sugars. And so this is, again, really like skewing my view towards like you actually have to go in and do the biochemistry, and like it's hard. Yeah, it's pretty tricky because like now we see a new peak, but then we got to go get this thing to fly on the mass spec, and like it's a crazy glycolipid. And yeah, but I mean, I agree. Glycosyltransferases, it's really an important unsolved problem. Like, yeah. Okay, the question in the chat is, do you think FlyX activity is changing in some of the PLED-like mutants? So I don't necessarily know the answer to this because I can't remember if Caulobacter even has FlyX. I think FlyX is either from E. coli or Vibrio. And I can't remember if Caulobacter actually uses that regulatory paradigm to control flagellar assembly. What I can say is that people in the past have studied PLED a lot, and there does not seem to be changes in the general transcriptional program that controls flagellar assembly. So if you all aren't familiar, in bacteria, like flagellar assembly is a classic model for studying transcriptional regulation because the genes are sort of expressed in waves, like the inner parts are expressed and then the outer parts and then the filament. And so people, when they first isolated PLED, went in and sort of confirmed that the flagellar assembly hierarchy seems to be intact. So I can't necessarily comment specifically on FlyX, but more broadly, I can say like we don't think that it's a flagellar regulation thing. We think it's a disassembly thing.
Well, let's go ahead and thank David again for the interesting seminar.